one very exciting new frontier in tissue engineering, and it's very, very early stages, and happening really starting at MIT, is a link not just between tissue engineering and systems biology, as I mentioned earlier, but a link between tissue engineering, systems biology, and the synthetic biology community. In synthetic biology, there's long been an effort on trying to engineer modular parts that would make primarily microorganisms carry out functions that they were never evolved to do originally, using a combination of forward design, putting in whole genetic circuits that make microbial systems interact with each other or make products that they wouldn't normally make but make them very efficiently, respond to light, have quorum sensing behaviors that they wouldn't normally have. There's been a tremendous set of advances in the synthetic biology community in harnessing design of genetic networks and the ability to implement complex programs of genetic networks within microorganisms. Recently, MIT has hired a number of faculty who are very prominent stars in this field and have moved into engineering mammalian systems, coming from the kind of EECS circuit design approach. How do you engineer a network to have a particular gain or to carry out an actuation function? Prominent is Ron Weiss and Chris Voigt. We have been fortunate to be funded by the National Science Foundation for a Science and Technology Research Center called Emergent Behaviors of Integrated Cellular Systems to figure out how do you design a biological machine using mammalian cells. How do you design something that, say, swims or moves and crawls around, detects a particular analyte, stops and does something. So using machine design principles derived from EECS and mechanical engineering and now applying them to complex networks of mammalian cells. So it's very exciting for tissue engineering because what we're going to learn is how information flows in complex interacting heterotypic cell systems. So one thing we really struggle with in tissue engineering is how to make a whole arrangement of cells that are very delicate do those things that we want them to do, to metabolize, respond to drugs, and so on. How is information really flowing? By applying principles of forward engineering of biological systems and understanding how your programmed genetic networks interact with endogenous networks, I think we'll learn tremendous things about how information flows and about how we can harness that to understand interactions of drugs and other things with the human tissues that we build in vitro. So I'm an associate director of this center, and it's a very exciting endeavor for the future of tissue engineering.